So just keep it in mind. It will be around November, December.
Okay.
And Dr. Mandakranta also would be requested to make a presentation on fruits and vegetables and the health of the poorest of the poor working on horticulture, something like that, because you are association with both with health and fruits and vegetables for a pretty long time. The topic will be yours, basically, but something to do with, this is basically focused on health through fruits and vegetables and nutrition. So this is the kind of a talk which is being envisaged. It is in very preliminary stages, but then I just thought I will, a formal request will come from Dr. Ayyappan, and then probably we will just take it forward, because Dr. Ayyappan has requested me to identify topics and persons and a lot of other things. It's more health oriented though.
Okay, sir.
Okay.
Thank you, sir. Can we go ahead with starting of today's session? Very good morning to all the participants, respected Dr. Krishna Kumar, Dr. Varghese, Dr. Patil, and all my colleagues who joined in this online Zoom meeting, particularly for two talks revolving around immunity. In the last 1 year, we have been listening very frequently the word immunity. That is where we choose this particular one. Being a biologist, that too into zoology or botany, or being a human beings, it is very, very pertinent if we are familiar with our own immunity system. In this regard, today we'll be having two talks, and followed by all of you will be the panelists, because more than the speakers, those who are attending, they might have more exposure for this immunity related aspects in either agriculture or insects or human beings. Dr. Mandakranta madam also is here. We'll be discussing relating to immunity in agriculture and those who are doing agriculture. For every organism, immunity is a must. In that regard, we'll go ahead with today's first presentation. And now I request Dr. Sriram, who will be talking on immunity in plants, plant immunity. Over to Dr. Sriram.
It is audible, sir?
Yeah, it is audible.
My respected seniors, colleagues, our former DDG Horticulture, Dr. Krishna Kumar sir, former Director of ICAR-IIHR, Dr. Varghese sir, and all my colleagues and friends, I am going to talk today about the plant immunity. And followed by this, Sridhar will be talking about the immunity in the insects, and Madam will talk about the human. So the outline it is a huge subject. The plant immunity is a very huge subject, and it is difficult to confine with everything in 1 hour, but I will try to touch upon our superficial view we will have about the plant immunity. Today we will be seeing about how the plant immunity is different from human immunity, and how the immunity works in the plant. It is mainly through the signal transduction, as well as the triggering immunity by two pathways. One is we call PTA in ETA. We will be seeing that in the later slides. Then the host pathogen interaction is more important here. How the plant interacts with the fungus, bacteria, and virus, and how the defense is induced. Then what is the parallel between the plant and human immunity? Is there any parallel? That also will be. And how to use this information, host pathogen interaction information, how we can use it for the management. This will be the outline. I will try to discuss, touch upon all those things in 30 to 35 minutes. The plants are affected by different pathogens, fungus, bacteria, virus, phytoplasma. They are either soil-borne, seed-borne, water-borne, insect transmitted. So the plant has to have the immunity towards all of these different types of visible and invisible microbes and the vectors. The diseases due to pathogens in human and plants, if you see, human also is affected by different microbial pathogens. Plant also is affected. But the human, we have the circulatory system So the antibodies are produced, and it is a different system. But the plant is not at all having the circulation system. There is no typical antibody production. There is no circulation system. So the immunoglobulin, IgG, IgM, whatever we talk in the real immunology terms, may not be applicable to the plant. But it has its own signal transduction pathways and the induction of defense mechanism. If you see the human, it is having both. We have both acquired as well as the innate immunity, but here it is mostly the innate immunity that has to be through this signal transduction. It has to be sent to the different organelles in the plant. And if you see the plant, as I told, we lack the plants like the mobile immune cells, the lymphocytes are not there. So that the circulation system is not there. So it is not possible for it to have the antibodies directly acting on that, but it has the indirect signal mechanism. The lymphocytes in the animal system, it clonally expresses the receptors, and it can move in the circulation system and detect the pathogens at different places in the body, animal body. If you see the research development in the plant immunity research, it is 40 to 50 years delayed compared to the animal system. Though there is a report in 1902 by Ward about the mention of immune response by the Bromus intermis to the rust fungus Puccinia dispersa, but much later only the science developed. There are similarities, analogous mechanisms happening in the plant and animal, but they are not identical. There are so far and yet so close. Like that is the expression given by Mekawa when he was comparing the plant and human immunity. One common thing is this salicylic acid mediation, we can say as an example. And some of the examples involved in the signal transduction, they are similar. They are having similar nature. But the main essence of both animal system as well as the plant system is the recognition and protection against the foreign, the non-self. That non-self recognition and protection is the main for both the systems with respect to the immunity. Okay. But when we come to the plant immunity, the resistance mechanism, it all starts, we started with the gene hypothesis given by Flor. He tried to explain the genetic of the resistance. For every resistance gene in the resistant plant, there is a corresponding pathogenesis related protein AVR gene in the pathogen. That was the thing. But later it was not able to completely explain many of the situations. Then came the God hypothesis, where there was a requirement for another specific protein to activate the resistance gene. So the God hypothesis came. Then this is not explaining all the resistant mechanism. It is applicable only for race-specific resistance or gene-mediated resistance only. It was explaining. There is parallelly one more phenomenon is happening, that is non-host resistance or non-race specific resistance. Those days in 70s, 80s, they were calling it as the elicitor-receptor interaction. Now the terms are changing in with the due course of time. So we will see what is that. What I mentioned about the elicitor and the receptor, that is the nothing but the PAMP triggered immunity. The pathogen, when it is falling on the plant surface, they secrete some molecules, and the plants perceive these diverse set of microbial molecules. We refer it as the microbial or pathogen associated molecular patterns. Why we have to use the MAMP? Why PAMP is not sufficient? It is only immunities against only pathogens. No, actually, the other microbes are also recognized as the foreign body. So the term MAMP also is used for the non-pathogenic microorganisms. MAMP we use for the pathogenic microorganisms. It is PAMP. For once this pathogen works against the host, then the host is not keeping quiet. It is having some receptors. So it recognizes that the foreign enemy has come. So it will start the recognition. This is where the immunity, the first starting point is this recognition. Once this recognition happens, the intercellular signaling happens, then the transcriptional reprogramming happens, the plant develops its defense in the cellular level. And another one is very important thing is that loss of cell wall integrity by the pathogen. Most of the fungal pathogens, especially the necrotrophic pathogens, they disturb the cell wall, and that cell wall integrity, once it is disturbed, that also gives a lot of signals. Plant perceives all these signals, and it induces the cell wall remodeling. It induces or it activates the different defense mechanisms like metabolites, mostly the phenol metabolites, shikimic acid pathway. So those things will start into operation. Then the antimicrobial action in metabolites will be secreted to restrict the microbial growth. There are a lot of examples for the PAMPs in the microbe or the pathogens. It may be the lipo oligosaccharides of the gram-negative bacteria or the bacterial flagellin. There are some examples, EF elongation factor 2, or the Pythium, Phytophthora, downy mildew, these oomycetes, they have the cell wall glucons or the glycoproteins. During the interaction process, the glucons released by the pathogen function as the PAMP. The host recognizes it as the PAMP and receives the signal and induces. Another one is the AX21, that is corresponding to the XA21 in the Xanthomonas. So there are different receptors in the plant also. Most of them are located in the cell surface, cell membrane located, they are located there only. So we will be seeing then a few more slides later, the cell wall is the battleground. So most of the receptors are present in the plant cell wall. Most of them are LRR, leucine-rich repeat receptor like proteins, or flagellin receptors, or the rice chitin binding protein, or in the Arabidopsis. So these are all the receptors that work, that they receive the signals of the PAMPs. Then corresponding to the PTI, pattern triggered immunity, we have the effector triggered immunity. So once the pathogen recognizes the PAMPs, and it induces the resistance, but the pathogen will not keep quiet. It will produce more effector proteins that will counter the plant defense, or sometimes these proteins exploit the host cell wall also in its favor. And these proteins are called as the effector proteins. And once they are recognized, the plant develops the immunity. That is the second line that we call it as the effector triggered immunity. This recognition of the effector is by the nucleotide binding leucine-rich repeats, NBS-LRR domain hosts a lot of genes in the plant that codes for the R genes. So this R gene mediated resistance comes under the effector triggered immunity, while the PAMP triggered immunity comes under the non-host or non-host specific resistance. So most of the R genes are located with NBS-LRR domain, and they have the LRR repeat that gives the specificity for binding interaction activities. Then not only the fungal pathogens, bacterial pathogens also secrete the effectors. Why I specifically mention about the bacteria is the bacteria have a specific secretion system. We call it as the T3 type 3 secretion system. I will show in some slides how it ejects the effectors into the cytoplasm of the plant cell and triggers the immunity. So in the fungal pathogens, it is through the haustoria or the other appendages through which the effectors are delivered. There are some examples, as in the PAMP, we have the examples for the AVR also. Pseudomonas syringae, a lot of AVR products have been reported. Cladosporium with the tomato, a lot of examples are there. There are many examples are there for the effector triggered immunity. But the plant also, after the triggered immunity, the plant receptors evolve and either recognize the obvious effectors that is already present, or the pathogen, they produce new effectors. So again, this also will co-evolve and give the evolving process continuously happening. That is why we see the boom and bust cycle, the breaking of the vertical resistance, the new race comes. That means it has got another new effect. It has acquired a new effector. The plan breaks it. That means it has evolved with another receptor for that. So it continuously goes on. This ETA process evolves over the time. So simply if you tell the PAMP, it is recognized by the receptor, and the PTA respond, the pattern triggered immunity happens. The pathogen, to overcome this resistance, releases the effector proteins. But plant will not keep quiet. It will fight with the effector, with the origin products. So origin is LBS LRR. So the effector triggered, the effector is suppressed, then we get the ETA response. This goes on continuously. It goes on. So the historically the terms used, elicitors and avirulence factors. Now we are using the word PAMPs, that is pathogen associated molecular patterns, and for the avirulence factors we are using the word effectors. Then came the zigzag model, that what I mentioned. This was proposed by the Jones and Dangle, that in the first place the PAMPs are recognized, then the PTA, through that the defense is amplified, then the pathogen produces the effectors, the resistance comes down, then the ETA, the effector is recognized, then we are getting the effector triggered immunity. Then the pathogen loses the effector due to mutation or something, or the NBS LRR genes in the plant, they counter the effector, then the ETA comes down, then we get the effector triggered susceptibility, then the pathogen produces new effectors, then so this continuously happening. This new effectors, this horizontal flow keeps going. That is what I was mentioning earlier as the co-evolution of the pathogen and the host.
Yes, सांपली दवाई लिए ना, इसके कोई सिकोरे हैं कोई सिकोरे?
Excuse me, somebody is taking.
देख सिकोरे जो हो.
Dr. Ramesh Kachwaha, please can you mute? So the pathogen encounters, this is the sequence what I have mentioned in the flowchart. I have mentioned whatever just I mentioned in the zigzag model. So in the PTA and ETA, the PTA is need not be host specific, it need not be race specific. Here also the antimicrobial protein.
Dr. Sriram, there are certain messages in chat box. I would request Sridhar to see, and see some persons are not able to hear. So Sridhar can look into the chat box and see if there are any problems to be addressed.
Okay.
Thank you.
Okay, sir. Please go ahead, sir. Let's read. Go ahead.
So in the PTA, we get the antimicrobial protein secondary metabolites, and in the ETA, effector triggered immunity, we get the R genes. Then we get here also defense gene expression. In both, we are getting the reactive oxygen species, hypersensitive reaction, everything. Here we may get the qualitative resistance or quantitative resistance with more number of genes. So effector triggered immunity is based on the floor hypothesis, gene for gene, and the PTA codes for the non-host resistance. Comparatively, the ETA generally is faster than the PAMP triggered immunity in the induction of defense mechanism. The hypersensitive reaction is much faster in the ETA. Or PTA and ETA, whether they are independent, they work separately, or they complement each other. Actually, earlier reports, if you see up to 2010, people were saying that ETA is different, PTA is different, and they are totally different. But we have got recently many literature reports suggesting that PAMPs and effectors cannot be separated. Same way PRR and R proteins cannot be separated. For example, I mentioned about the exam gene in the Xanthomonas resistance. The same receptor is working for PAMP triggered immunity also. In the ETA triggered immunity also, it is working as the receptor. Same receptor is working for both the immunity systems.
That's Sriram. Please unmute and go ahead. I have muted everybody. Dr. Sriram, please unmute and proceed. Dr. Sriram, please unmute and proceed. Hello?
Now I am audible, sir.
Okay. I have unmuted everybody. That's why this interruption. Please carry on. Okay.
So now we cannot differentiate the PAMP triggered immunity and effector triggered immunity in some cases. We will see one or two examples also. The recent literature, that is 2 months back only, it is yet to be published. It is in the early stage only. And Pruitt et al. In 2021 March, they have studied with the Arabidopsis and Pseudomonas syringae system. So here, if you see the first PAMP triggered immunity happening, it is supposed to induce the one particular receptor in the host plant, and it has to increase the respiratory burst oxidase protein. But the pathogen releases the effector, it suppresses it. So the defense is ineffective. But in the presence of the ETA, that is NBS LRR domain in the plant, it has the gene and it interacts with the effector, and it triggers again the same enzyme which is supposed to be triggered by the PAMP. That is again the RBOHD gene only. So it triggers again the PRR related defense mechanism only, receptor only. So we cannot now separate PTA and ETA. They are complementary to each other. Both are required. That is why in many cases we are not able to say that gene for gene hypothesis is holding good for segregation of the genetics of inheritance of the resistance. There are a lot of biochemical mechanism happening in the inside the plant, both the PTA and ETA. Moreover, in the NBS LRR domain also, there are many genes. So one is activated, the other gene also gets activated. So it is not one we were expecting for the expression of only resistance to only one race, but it gives the resistance to 2-3 races also possible. That is because of the togetherness, the activation of the many genes in the NBS LRR domain at the same time. So to understand the resistance, we need how the pathogen and host they interact with each other. So the breaking of the cell wall integrity is the main thing. So as I mentioned in the beginning, the cell wall is the battleground where the pathogen and plant they fight it out, and the determination of the resistance or susceptibility comes from at this point only. When the pathogen meets the host cell wall, the decision of whether it is going to be a resistant reaction or susceptible reaction is determined at this stage only. And further, the lifestyle of the pathogen is important. For example, fungal pathogens, if you take two types are there, one is the necrotrophic pathogens, another one is the biotrophic pathogens. If you take the necrotrophic pathogens, they kill in advance the host cells, and they use the cell cytoplasm material for their food. So they have to kill the plant cells. So they generally secrete a lot of cellulolytic enzyme, that cell wall degrading enzyme, cellulase, polygalactronase, hemicellulase, pectin methyl esterase, acetyl esterases, like that. The plant uses once this degradation is happening, lot of oligomers are released. These oligomers act as the PAMPCR, and the plant has in its cytoplasm, in the cell wall, the plasma membrane, if you see, these oligomers go and bind with the receptors. Most of the receptors are present on the plant membrane, plasma membrane. For example, one here is the WAK1, wall-associated kinase 1. So the oligomers find, once it is binding with the WAK1, then the defense response increases. The plant, to defend itself, it produces the polygalactronase inhibitor protein, PME inhibitor protein, or xylanase inhibitor protein, peroxidase, and extensin. And if you see simultaneously, the cell wall components, the integration has to be maintained. To reinforce the cell wall, it secretes, it produces a lot of callose. And this calcium mediated cross-linking happens. So all this ferulic acid cross-linking, all these are happening in the resistant plants. If the resistant plant is there, that means there are some receptors in it that has received or perceived the PAMPs and defense mechanism has happened. In case the resistance and susceptibility is determined based on the game who has won this war at the cell wall level. If we see in the susceptible one, there will not be any host-induced defense mechanism. The degradation will continuously go and happen, and the cell will die, plant cell will die. But in the resistant one, lot of biochemical events happen, and the cell wall integrity is protected. Different defense enzymes are produced. In some cases, the reactive oxygen species and cell wall, that hypersensitive reaction may cause the necrosis, or the programmed cell death also can happen and limit the invasion of the pathogen. This is with respect to the necrotroph pathogens. What happens with the biotrophic pathogens, where the fungal pathogens, for example, powdery mildew, downy mildew, or the rust pathogens, they don't want to kill the host cells in advance. They want to be there, sporulate, complete their life cycle. Once they are completing all that, secondary spread is happening. Once they have completed their requirements, then only the plant is allowed to die. So till that time, they want to keep the host cells alive. That is why if you see the powdery mildew affected leaves, they won't die immediately. Necrosis will not be there at the fag end only, it will be drying. So the fungus, it produces the infection peg. The infection peg goes inside. At time, the plant is defending it with the peroxidase extensins or the cellulose, callose, and all other material. For this also, the pattern recognition receptors are required. What happens in the susceptible variety as well as the resistant one? If the resistant plant is there, in its plasma membrane, there will be a lot of callose deposition happening in the cell membrane. Callose, it prevents the infection peg going inside and forming the hostoria. Reactive oxygen species is there. Then cross-linking of the cell wall, either the cellulose, hemicellulose, pectin, interlinking with the lignin, everything happens. So the cell wall is reinforced. Resistance happens there. The infection peg is stopped there. It is not penetrating inside the host cell. What happens in the susceptible one? Here, the infection peg is not stopped. It goes inside and forms very good hostoria, and it interacts with the host cell, manipulates its requirement, and takes all the nutrition required for it through hostorium. The fungus flourishes very nicely. So if the resistant variety is there, that means it has a receptor that triggers the resistant mechanism. If it is susceptible one, there is no receptor, so there is no stopping of the infection. So this is the one. Together, if we see the susceptible one, it is not able to stop the penetration of the infection peg, and hostorium formed, and the pathogen flourishes very nicely. We come to the bacteria. So bacteria also have a lot of PAMPs I mentioned earlier, flagellin, and it triggers different mechanisms like fungal cell only. Lot of enzymes are produced by the plant in response to that if it is having the receptors. But when compared to the effectors, here it is slightly different compared to the fungal. The effectors of the bacterium are secreted inside the cytoplasm of the plant through a specific apparatus called a type 3 secretion system. Even a very good designer engineers cannot design such a very nice delivery system. The bacteria has its own type 3 secretion system. The effector proteins are very safely passed through the pilus system, and it is delivered inside the plant host. In the effector proteins are secreted. This we can see in some, this slide explains both the PTA and ETA. There are a lot of videos are also available how the effector proteins are delivered inside the cytoplasm of the plant through the pili system of the bacteria. We can see nicely the pili system, it goes inside the host and it releases the effector proteins. This type 3 secretion system is very well established, evolved over the time, and it helps the bacteria not only for the pathogenesis related virulence factors, but in later, if the host triggers these proteins, it develops the resistance. There are 6 types of secretion systems in the bacteria. In the gram-negative, the type 1 and type 4 are present in both gram-negative and gram-positive. In gram-negative, all the 6 are present. So this type 3 secretion system is very, very important to understand. Even if you are able to develop some possible antibiotics to interact with this effector protein, that will be of great use when we study the bacterial resistance. When we come to the resistance to the viral diseases, both dominant gene mediated or gene mediated resistance through NBS LRR domain are also possible. Then lot of examples, many examples are with the recessive genes. That is especially the 4E, 4G, that is translation initiation. In next 2 slides, I will mention about how these translation initiation factors are useful to stop the virus multiplication, and we can exploit it for the virus resistance. Though the other mechanisms are the RNA silencing can be used, this R gene mediated can be used, recessive inherited resistance can be used. So either the overexpression of the translation factor can be used, or the genome editing or gene silencing we can use for the translation factors. These are all possible ways of getting the resistance to the viral diseases. So there are 2 mutations are required in case of the resistance to the viral diseases. One should be a passive, another one is the active. The passive one I mean is that it renders the pathogen unable to hijack the host cell wall mechanism to multiply. That is the loss of the susceptibility or the host resistance. The plant is not allowing the virus to multiply. The factors that are present in the plant, that is not going to allow the virus to replicate. Another one is the absence of dysfunction of a negative regulator of plant response. In simpler terms, the defense mechanism is not stopped. Earlier, there are some mechanism that was suppressing the defense mechanism against the virus. Typical example has been reported in rice yellow mottle virus, RYMV2 virus. This has been the first due to the loss of function mutation. The function has been stopped. The suppressor is no longer going to stop the defense mechanism. And compared to all this, depending on the mutations, the translation can be better used. The translation elongation, that elongation initiation during the translation is very important. The virus goes inside the plant, the virion is uncoated. It depends on the translation. So here lot of elongation initiation factors are playing major role. They are called as EIF family of proteins. They are required for the virus to multiply and go out. So if we are attempting RNA silencing, we can target these proteins for our CRISPR or any other gene silencing program. This translation mechanism, if we are interfering so that the virus is not replicated, not the elongation initiation is stopped, that itself is sufficient to have the resistance against the viral diseases. So there are a lot of information are available with the host to pathogen interactions, how we can utilize it. One is harpin genes, especially mentioned about the type 3 secretion system in Pseudomonas syringae. They have come with the harpin protein. If we are spraying in a low quantity, low concentration, it can trigger the defense mechanism. This has been reported in China and some other countries. In India also, at Hyderabad, Pudili Apparao, Dr. Pudili Apparao, he has made a lot of progress in this aspect, and they also have developed harpin like proteins related products they have developed at the Hyderabad. Then, as I mentioned about the EIF related, this family related proteins can be used for the virus disease resistance. There are many translation factors, different species they have used for the resistance against different viruses. This can be exploited. How we can exploit this? We can use this in the CRISPR, we can use in the RNA gene silencing, and this can help in getting the resistance to the viral diseases. For fungal diseases in our lab, my PhD student, she worked on the transcriptome with respect to the rose and powdery mildew interaction. We identified the resistance gene analogs and the proteomics work and transcriptome work. We could identify one particular gene, that is RPP13. This resistance gene, this is present in the ICAR 13-4. Dr. Tejaswini madam has identified this. This is resistant to powdery mildew. Compared to the confetti, which is a highly susceptible variety, this gene is not there. And we have studied proteomics. There are 3 different proteins also. So how can we utilize is that these can be used in the marker-aided selection. We can use these for the marker-aided selection. We can either use this for breeding work as well as we can use it for the marker-aided selection. So this way we can use this information generated in the host pathogen interaction for our disease management. So I mentioned about the parallels between the human and the plant immunity. Recently, 3 proteins identified in the human immunity. They are almost structurally and functionally similar. Proteins have been identified in the plant also. HMGB1 corresponding, that is related to the inflammation related disease in the human. We have in the plant HMGB3 that interacts with the salicylic acid. They are almost similar. And the same way, another protein is the mixed lineage kinase domain-like proteins. This is present in the vertebrates. Mekawa et al., they have identified it in the plants. So, though they differ in physiology, habitat, and the different requirements, the plant and the animal, they try to keep off them with the help of receptors that are evolved independently in different mechanisms. So if you see the resistant mechanism, it is very difficult to explain it in one session. We may think that the resistant plant is very cool and it is very happy, but a lot of storm of biochemical events are happening inside the plant that we may not be able to visualize. So much the storm of biochemical reactions are happening inside the plant. I try to touch upon the different mechanisms, how it is in the plant with respect to the immunity. Thank you for the opportunities.
Thank you, Dr. Sriram, for our excellent coverage of plant immunity. Now we'll have a discussion for 10-15 minutes related to plant immunity. Now the session is open for discussion. Dr. Krishna Kumar, sir.
I would ask if I would give opportunities to all the others, and then I'll come back because it's
okay. Others, please, and we'll restrict straight questions for to the extent of maximum 15 minutes, and you can reserve the questions at the end of after insects, after listening to human beings. Also, we'll have the general discussion later. Now, I request any straightforward questions regarding plant immunity or your contribution from your side. Also, you can share some information on plant immunity if you have.
Subharan is raising question. Good morning, Subharan.
Okay.
Good morning, Sriram. Good morning.
Welcome. Very good morning, and
good morning to you all. Good morning to you all, sirs. It was a wonderful talk. Actually, it is a break of boredom during this lockdown period. Wonderful talk, Dr. Sriram. I have been having a doubt for a long time, right from your PhD days when you were doing your work on pathogens-related proteins.
Subharan, can you please speak slow?
Sure, sir. Sure, sir.
Speak a little slow because the purpose of it is others may not be able to follow what you're asking.
Oh, I'm sorry, sir. I'll be slow, sir. And Dr. Sriram, I just have a doubt. You have been speaking about ETA and PTA. PTA first starting and then ETA coming, and then you finally ended up in a zigzag cycle. I was just trying to have a doubt in my mind, and I wanted clarified for a long time, and I thought this is the right time. When such activities of PTA and ETA are happening in the host, there should be some metabolic cost involved. And in such a case, how is the plant going to compensate by itself? Or do we need to give an external supplement? Because just like human analogy, drawing a human analogy, when you are sick, of course, the immune system tries to fight, and we do need some supplements to keep ourselves going because there is some metabolic cost as far as the immunology is concerned. Is there some such thing that is happening, and do we need to supplement with something externally to keep, especially I was very interested in that zigzag. So when cycle is happening.
Yes, in case of the effector triggered immunity, especially the origin mediated resistance, there is a trade-off happening that the yield component is getting reduced because the energy is diverted towards the biochemical mechanism to fight against it. So that is why once these origins are identified, we have to go for the crossing with the better yield. That is again, it is required. Only if you take only that particular resistant material, yield we have compensation is not, yield is compensated. We have to do the back crossing with the high yielding varieties. Yeah, you are correct. It is, there is a metabolism taking a lot of energy. Yield component is reduced.
Thank you. Thank you.
Supplement is required.
Thank you. Thank you.
But not with the PTA, especially with ETA reports are there.
Okay. But can we think of an external supplement rather than going in for the genetic basis?
External supplement, this is the Harpin is the external supplement. And one more thing, what we used, I have not presented here, the Phytophthora produces the glucan. From its cell wall, we can extract the glucan, and we can use that elicitor as the screening material to identify the resistance sources. But application of that is, it has to come to its own time. As you know, from the trichoderma, we have identified the ISR inducing proteins, some small one protein that can be externally applied, but cost involved, everything is very high. Fantastic. External application.
Good. Thank you. Thank you very much.
Okay, next.
Dr. Sriram, I have a couple of small pointers. The pathway of whether it is hydrogen peroxide or whether it is followed by dismutase or the series of enzymatic and non-enzymatic response pattern is more or less similar to, it follows more or less the same pattern irrespective of the kind of normal pathogen entry leading from hypersensitivity to suppression of the entry of this thing. But what we have not understood in plant immunity is the specificity that is associated probably with regard to each of the pathogen. The response pattern, and if you follow the pattern is more or less similar. It follows the same pattern from one end to another end. First it follows the hypersensitive reaction, then it suppresses through enzymatic and non-enzymatic process. At this particular point of time, I would like, while we are going too much deep into the biochemical, is it time to look into the biome associated with the host and the pathogens and their role in fighting? Because I find that even in pathogenesis, it is not an one-to-one reaction, but it is a kind of the community associated enzymatic, non-enzymatic processes that are happening in the rhizosphere or in the whatever the surface of the leaf or whatever it is contributing. Probably that has not been so well documented that at the end of the day, like double-stranded RNA, what we are looking and what is happening may be a little bit different. And I think, in my opinion, that needs to be a much more focus. That probably vastly explains as to, in spite of the pattern being the same, why a number of pathogens are not able to invade.
Yes, sir.
The number, that's one observation. You respond a couple of points, I'll tell, and then. The second one is suppression of the entry of the pathogen and silencing of the pathogen. They are two different phenomena. And we cannot mix the suppression of the pathogen that occurs at the cell wall level. And I was a little bit confused when you said cell wall. Did you mean cell membrane? Or when you meant cell membrane, did you mean cell wall? Because the enzymatic process to dissolve the cell wall, but the epitope ligand binding, in my opinion, takes place at the cell membrane level. So we, in plant system, we have to be very clear. The pathogenesis from cell wall to cell membrane there are a lot of interfaces that need to be understood. The third one is entry of any pathogen into the cell membrane takes place by two methods. Either it is by clathrin mediated endocytosis or it is by facilitated endocytosis. Now, we have not really understood the lock and key mechanism between the pathogens and the epitopes associated with the host membrane, and we are brushing everything with a very general carpet. The aspects that I wanted to tell you is, in the case of viruses, does the plant recognize it as self or non-self, considering that it is a living or a non-living entity? This is what is your opinion? I'm not asking for when it comes to the virus, somewhere does the organism treat it as self or non-self or just another protein? That is a little bit a very tricky issue. In the case of viruses, the assembly of viruses is not in one go. Golgi operators get separately assembled, nucleocapsid protein gets assembled separately, and all of them have by a portion of the reassembly that takes place, then basically it goes out of the cell wall into multiply. My last question is, the resistance to replication was resistance to movement of the viruses. What are the mechanisms involved? I have many questions.
Yes, sir, I have noted down.
I'll stop you.
I have noted down, sir. Sir, what you have mentioned, the microbiome surrounding the rhizosphere place, actually there are now the usage of the second genome of the plant. That is nothing but the microbes surrounding the plant. They are the second genome of the plant in the phytobiome. The microbes surrounding the plant that play a major role. That is why there is in phytopathology, there is a separate journal also. Supplementary material is coming as the phytobiome. So a lot of information is coming from there. We are yet to capitalize on the progress made in elsewhere. The phytobiomes, the microbes surrounding the plant, if we understand how they prevent or help the pathogens, in fact, we can understand, sir. I agree with that, sir. Then second one, you mentioned about suppression and silencing. Silencing I was using for the virus related RNA silencing. Suppression is for the fungal and bacterial diseases, sir. Here in viral cases, I was not. We are not using the suppression. There, the gene silencing is playing major role with the viruses, sir. Then regarding the plasma membrane and
what if there is a hypersensitive reaction to a virus infection? Is it suppression or silencing?
Sir, in the hypersensitive reaction is due to the when the highest form of resistance is the highest form of susceptibility is the resistance. For example, programmed cell death. It is like a programmed cell death.
Occurs, I thought it is not silencing.
It is not silencing.
Yeah, that's what I meant.
It is the virus multiplication is stopped because of the highest form of programmed cell death.
Okay.
And then again, regarding this plasma membrane, all the receptors are present in the cell membranes.
Has there been any in-depth analysis of the role of lysosomes, which are involved in cell death, in relation to the hypersensitivity between and resistance?
Yes, sir. Lysosomes play major role in the programmed cell death. In-depth work has been done with respect to fungal pathogens and bacterial pathogens for the PCD, sir. It is there.
Okay, I'll not take much time. Let others also ask.
Then you mentioned about the virus, whether it is self or non-self. Since it is having the nucleic acid and it is triggering its replication, I think the plant cell may be recognizing it as the non-self, not as a external molecule. It is not a molecule. It may be treating as a non-self. That is why it is fighting. Or otherwise, there may be some exclusion mechanism to send it out.
Fight for any protein also.
Right. So
when it fights for a protein, it is not self. Anyhow, that's a debatable issue. Let us leave it at that.
Okay. Thank you, sir. Thank you. Dr. Meena.
Sir, one more question. Excuse me, sir.
Yeah.
Sir.
Okay, please go.
Good morning, sir. I am Anshuman Khandwal. I am assistant professor in plant pathology. Sir, I want to ask one thing, that whether there is a clear-cut difference between immunity and resistance in plant pathology.
Plant pathology, we have to use the word resistance based on the plant microbe interaction. It is the resistant mechanism. Immune means there is the typical, in the first slide I was using the word, the typical immunology related terms are not applicable here. The immunity means, for example, the Phytophthora infestans spores you take and put it on the rice. They don't recognize each other. That may be, we call it as immune. Or when we put the rust pustules on the potato, they don't recognize each other. That may be immune response. But resistance, we are interested in the resistance based on the host pathogen interaction. They recognize each other. The recognition word is more important.
Here the races or the strains comes into play.
Yes. Yes.
Okay, sir. And one, sir, for my information, I'm asking, sir, a few months back, I have attended one of your talks. It was on trichoderma formulations. So I want to ask you, sir, one thing, that what is your actual field of research, sir? I am interested to know.
Up to 2014, I was working at NBAIR now, S12 Project Director of Biological Control or National Bureau of Agricultural Important Insects. That time I was working only on biological control. Now,
sir.
after moving to Indian Institute of Horticulture Research, I am working both on the biocontrol as well as host plant resistance. Both I am working.
Host plant resistance, that rose powdery mildew interaction,
yes,
means
that is our work.
Only on that, or other interactions are also there?
We are working on fruit crops also. In the Goa, we have identified some rootstocks that can give resistance to the wilt disease. And in other diseases also, we are working for the resistance breeding. I am working as a collaborator.
Thank you, sir.
In the breeding programs led by
Yes, sir.
the breeders, I am working as the collaborator.
Yes.
Okay.
Thank you,
Okay. Thank you. Next question.
Good morning, everyone.
Dr.
Dr.
Meena,
Sriram, that was a very nice talk. Really enjoyed it. I had a question about whether anything is known about plant immunity to prion-related diseases.
So far, I have not come across the literature. Maybe it may be there. It may be my default that I have not come across. Maybe there, but I have not seen.
Okay.
I have not come across.
And the other question is, in case there are two highly competent pathogens which co-infect a plant, what decides which pathogen takes over? Or does the plant win in this case?
No, that is based on the interaction with the receptors. If the plants have the receptors, they trigger the immunity. If the receptors are not present, then the pathogen wins. But in the co-evolution, it is a continuous evolving process. The plant develop the recognition factors to identify the effectors, and the pathogens also come with the new effectors. It is co-evolution.
Yes, but wouldn't the two pathogens compete, and like wouldn't that dilute the infectivity?
You mean two pathogens?
Yes, two extremely competent pathogens. If they just co-infect a plant, would they compete with each other and then just dilute the infectivity, or?
No. Their recognition mechanism is different. For example, if you have one tomato leaf is there, powdery mildew has started coming. So already the defense, whichever comes first, it induces the defense mechanism. The second coming pathogen may have less probability. If both are coming with the same quantum of inoculum and same quantum of virulence, then whichever is recognizing the first and whichever is able to trigger the defense mechanism, that will win. The second one, there are a lot of reports for the reduced infection by the second pathogen when already diseased plant is infected with the second pathogen. Literature is there. The first one which is inducing the resistance much early. For example, the phenylalanine ammonia lyase is induced within few hours of infection. So whichever is producing that, initiating the process, that will win.
Okay. Thank you so much.
Dr. Sriram,
Sir, excuse me, sir.
There is a concept called niche concept, where two organisms cannot occupy the same niche at the same time. So when there is a two virulent pathogens really come together, whichever is the dominant pathogen will not suppress the other pathogen for its own advantage. So ultimately only one of them will remain.
So, sir, if in this case there is one biotroph and an necrotroph, so if the biotroph occupies first, then the necrotroph will get the advantage from the biotroph as well as the plant. I mean the dead host.
But the necrotroph will have upper hand because it kills the cells immediately. So the biotroph will have the lesser chance compared to Phytophthora infestans and the powdery mildew. The Phytophthora has more chance because it kills. Without the host cell, biotroph cannot survive.
Yes, sir.
So the biotroph will have more chance than the necrotroph will have more chance than the biotroph.
No, sir. If the biotroph has already occupied and there is a necrotroph waiting there, so the host will be weakened and ultimately the necrotroph will get the upper hand, means due to the advantage from the biotroph and the weakening of the host. I was asking.
No, but in case of the necrotrophs, the
Yes,
cellulolytic enzymes are much faster and they degrade host cells, and these signals are transferred systematically. So the progression of the disease is much faster. That's what I wanted to say.
So
Okay.
if sir in plant pathology scenario we will means if plant diseases scenario we will see. So which is more destructive, whether it is necrotroph or biotroph?
Both.
More, sir. Means comparatively.
That we cannot say based on the variety, based on the virulence of the pathogen, we have to decide.
Okay, sir. Okay.
Okay. Okay. Thank you. Any supplementary information by any other audience? That's Sai Prasad, Manmohan. I could see that anybody else. You can just within another 5 minutes. We'll move to the next talk. Then we will continue the overall discussion.
Any information on black fungus, which is of common interest to everybody? It is both a plant and an animal interest right now.
Right now, some injection is available for that, sir.
No, it's okay, but of interest.
It is.
No, that will come later. No, that will come later. That discussion will come later.
Yeah, that is also in today's syllabus. It will come.
Yeah. Sridhar, I just want to make one or two quick remarks.
Okay, please.
Dr. Sriram, it was very informative, and I learned a lot. There are many new concepts. I just wanted to know, Krishna Kumar also was mentioning about the biome concept. Now, it is been told that when the ARCA microbial consortia, is sprayed, which is not actually acidal in real respect, but what it induces a lot of resistance, and pomegranate nodal blight is considerably reduced. That is one point. And if there is a mechanism, is it something to do with what you were explaining? Number one. Number two, I also have I'm one of the IMO consultants, and the farmers, one group of farmers who have 6 acres of tomato, they have told me that Panchagavya is very good for the blight in tomato. See, I was not a proponent of these kind of sidles. Panchagavya to me appeared very funny to me. But when I actually involved with him in doing this, and he showed me, Mancozeb sprayed about 2 acres beyond tomato, and this Panchagavya, the alternaria was suppressed, in that plant. So, and then the microbiologist explained that there are several kinds of microbes in this Panchagavya. It was fermented in the farm only, in the person's farm, which is one of the IMO requirement. Now, there also what is happening, something is taking over. You mentioned about that. But actually, I'm yet to understand what exactly is that mechanism. Do they occupy the cell wall, or they take over the cell wall, or they become receptors, effectors? How does it happen? These are the 2 points related to that. And then I would like to know if any bioinformatics has been involved to come out with a model which would suggest to a breeder how to breed a resistant or produce a resistant plant. I mean, these 2 issues. Thank you.
Right.
First one about AMC. So it has 3 bacterial cultures. Actually, the main function is the avoidance of the disease. The competition for nutrition and space by these bacteria, they occupy the space first. Once it is either in the root zone or that is avoidance, sir. As a basic principle of the biological control, it works as the avoidance. It is not
Is it
inducing
inside the cell? Is it
No, sir,
inside the cell
outside.
on the cell wall or just external?
Sir, as we have, it is external, sir. It is preventing the host pathogen interaction by competing with the nutrition and this space, and so that the pathogen is getting less chance to come to that. Sir, second, you mentioned about the tomato case. It is nothing but instead of PAMP, I will call it as MAMP, microbial activated molecular patterns, MAMPs. So, for example, if you take one child from a slum area and Manyata take part to some apartment, very posh guy, 2 boys, you take All India tour and come back, the first fellow to fall sick is the Manyata Park fellow, because he has not been exposed to the microbes. Same thing happens here also. When you give the Panchakavya or whatever, those microbes are treated as the non-self by the plant, and it responds to that, but not in the hypersensitive reaction, not in the gene for gene hypothesis response. It is not host pathogen, but as a external microbe, it gives the resistance. If we remove those microbes, the slum boy also will become the susceptible. So same way here, the MAMP is playing into roles. Then you mentioned about bioinformatics, sir. The last some reports I mentioned, the similarity between the plant and human, it is purely based on the bioinformatics work. What they were doing, what is the proteins present, what are the genes present in the human, whether they are present in the plant, they were searching for that. Then only they could identify MLKL protein. So that they produced protein and they tried to control the gray mold, that is Botrytis cinerea. So bioinformatics plays, it is going to help us long ways.
Sridhar, I have one small, thing to ask Sriram.
Yes, sir.
Please, little bit describe steroid, microbiome, and black fungus for the benefit of the house. Steroid,
right,
biome, and the incidence of black fungus.
Steroids help stop the immunity in the human being. So generally, even in the COVID patient, even I was affected by the COVID last year. So the doctor did not prescribe COVID in the first instance. He told it suppresses all the immunity, and once the immunity is suppressed, the fungus takes over. That is why the doctors are also telling the steroid recommendation dose people have to follow. Once the steroid level comes, then the fungus takes over. The steroids, they suppress the immunity in the human being. And that is why the black fungus, they multiplies.
What is the equivalent in plant system?
Plant system, we don't have steroid types, sir, but we have the different phenolic compounds for suppressing the fungus. We don't have the equivalent to, actually the fungal pathogens need steroids, except the Phytophthora. All the other fungi, they need sterol in their biological functioning.
But in human, are required. See, sterol requirement is there across all living things. Whether it comes through which source is only the, it can come through beta cytosterol or it can come through many things. So sterol is an inbuilt requirement of any organism, but not necessarily as a part of nutrition, because they can reconvert it. My only this thing is why I asked you was, do the many times the fungicide act de facto steroids?
No, sir. Fungicides are not acting as steroids.
By sterilizing the soil.
Actually, this triazole group of fungicides, they are affecting the synthesis of sterol, but not as the steroids.
Okay.
All triazole fungicides, they inhibit the demethylation of this sterol biosynthesis, especially the ergosterol.
Pomegranate nodal blight, excessive use of Bactrin is many times attributed to the incidence of nodal blight. Okay, anyhow, we have, I think, run out of time. Sridhar, you have now.
Okay, we are running out of time. We actually put 1 hour for this talk, and we can continue this one.
But let us give a big hand to Sriram for a good talk.
Yes.
Thank you, Sriram.
From all the participant side, I'm conveying heartfelt thanks to Dr. Sriram for agreeing to give this talk in short notice. I thank all the participants. Now we'll move to the next presentation on same topic, immunity in case of insects and human beings, how it is. Now I'll be sharing this talk, this presentation. Is it visible?
Yes, visible. It is visible. Go ahead.
Welcome to all the participants for this second lecture on immunity in insects and insects versus humans. This will be my first attempt to introduce this topic from my side. Many of you may be already much familiar, but I took some time, I spent some time to come together because of my curiosity I developed last 1 year. What is this immunity word is coming very repeatedly, almost daily in newspaper. There is a without this immunity word, there is a no newspaper, daily newspaper. Then being an entomologist, how it is in an invertebrate, in insects, how it is there, and in humans. For all of us, it is required how to understand that immunity in case of humans. Comparatively, now we will see this is the slide. I took it from Professor Hoffman, who got the Nobel Prize in 2011 for physiology and medicine. Particularly, he worked on the insect Drosophila. Drosophila is the insect which gave 6 Nobel Prizes for different works till now. And in his first narration, what he told, somebody asked that question, what was the purpose of taking an insect for studying this immunity? His answer was, insects account for 80% of all living species on Earth, and they annually destroy 1/3 of human beings, and they put 1/3 of humanity and livestock at risk of bacterial, fungal, viral, and parasitic infections via other vector capacities. And mainly, they are particularly resistant to infections. That is what made them interested to study on immunity. As we know, these insects are affected with the different microbes like protozoans, viruses, and fungi, nematodes. Different microbes will be affecting these insects. And till they gave the theories of how this immunity is happening, how it is initiated, there was no clarity, though that immunity word is existing. And that's how 3 persons shared that Nobel Prize in 2011 for this particular immunity studies that we will see in the coming slides. Coming to the type of immunity, that Sriram has given a clear differences in case of immunity, and here and there how it is different in case of plants, and even try to compare in human beings. If you see, in case of insects or human beings, we will take this as either invertebrates or vertebrates. Invertebrates for the take for example insects, and vertebrates will be taking the example of human beings. The total immunity broadly can be divided into 2. One is innate immunity, which is present in all animals, insects as well as human beings. Not only human beings, all the animals which are present. Even the example what we can take is plants. What we listen till now, these plants are also have the innate immunity. Here, what is the major difference between this innate and adapted immunity is, in case of innate immunity, recognition of the traits shared by broad range of pathogens using small set of receptors is taken place, and the response is very fast. Suppose some pathogen attacked, response will be very immediate, but that particular organism or animal won't recognize when it comes again, second time. That is innate immunity, and it comes from by birth. That is the thing. And what are the different innate mechanisms? If you see, while telling, you can keep in mind barrier defenses or internal defenses. You can keep both insects and human beings in mind, like skin. Suppose in case of insects, it is a cuticle. For us, it is a skin, largest organ in our body, and in terms of area. Then mucous membranes or secretions. These are the barriers. And what is their role? They won't allow these microbes from entering inside the body. Suppose by chance it has entered inside. Internally also there are several defenses, both in case of human beings and insects. That we call it as phagocytic cells, and natural killer cells. They are not in insects but present in human beings. Antimicrobial peptides, they are present in human beings as well as insects also. And inflammatory responses, they are very specifically present in human beings. These are the internal defenses. That way, innate immunity can be defined as barrier defenses and internal defenses. Details we'll see. Coming to the another aspect, what we'll be seeing now is adapted immunity. What is adapted immunity? Adapted immunity is here we need not to go for much discussion. We'll keep only COVID as an example. Once COVID comes, suppose last year some of us got COVID. Now already we are adapted. It is there. Suppose if it is not, we have not got that one last year. We got the vaccine and the antibodies are multiplied within our body. That is also adapted immunity. Here, this particular immunity is not present in insects. That is what we have to see. But there are few studies last 2 years. There are some 1 or 2 examples of are insects also have this adapted immunity. Like that, studies are there, but they have to be still confirmed. One example of development of adapted immunity against Beauveria bassiana and Drosophila, it was studied, and it is to be fully accepted. And otherwise, this particular adaptive immunity is the major, its presence is in jawed vertebrates or mammals. Here, what is the broad criteria of giving adapted immunity? One is humoral response, another is cell-mediated response. Humoral response means whatever body fluids we have, it may be a lymph glands, and what are the different glands we have, we'll see in the coming slides. What that body fluids in the form of development of antibodies, how they are defending against infections, that comes as a part of humoral response. Another part is cell-mediated response at cellular level in the form of delineating or removing the through cytotoxic cells. How we are getting protection from these external microbes, that is covered under cell-mediated response. Here in adapted immunity, unlike innate immunity, recognition of traits is specific to particular pathogen. If we have developed resistance to COVID-1, antibody 1, that is resistant to that particular strain. If it is a new strain, new antibody is required, and that way it should be very specific to that species or strain. That way it will go. This is in brief introduction about types of immunity in case of insects and human beings. Here, when you are trying to understand this immunity, either in insects or human beings, we should recollect these 3 scientists who got Nobel Prize in 2011 for their work on immunity only. Exclusively, their work is related to immunity. Why they got this particular Nobel Prize in 2011 was shared between 3 scientists. First one is Bruce Butler. He got 1/4 prize. His contribution is he has identified toll-like receptors. What is that? We'll see later, related to immunity. Middle person is Dr. Hoffman, whose slide I used initially. He got 1/4 prize. Why he got is, he did his work on Drosophila, and he identified how this initiation of innate immunity is starting, or what is the procedure of starting of immunity in case of insects. He gave that theory. For that he got that Nobel Prize. The third person is Ralph Steinman. In 1973 itself, he identified very important cell in our human body, dendritic cells. Dendritic cells are very important. which is almost like a connecting link between innate immunity and the adapted immunity, that is very much essential in the formation of memory in terms of this immunity. For that, he got half the price went to him. This third person, for him, it is mainly the adapted immunity. Other 2 persons, it is for the innate immunity. Overall, these 3 scientists shared the Nobel Prize for their contribution towards understanding immunity. Coming to the to sum it up, what we have seen till now, innate immunity is response is non-specific. Adaptation is opposite to that. And in innate immunity, exposure leads to immediate maximal response. It is a lag time is required in case of adaptive. In this particular innate immunity is both cell-mediated as well as humoral component. That is same in both adapted and immunity. There may be a difference in the names of peptides or name of the chemical, but the broad pathways are similar. What is that we will see. In case of innate immunity, we will see first in insects, they have cell-mediated procedures like nodulation, encapsulation, phagocytosis. This is very common. In case of humoral components, antimicrobial peptides, when some microbe has attack, body starts producing antimicrobial peptides, which will be acting on the microbes. And the activation of the prophenoxyl oxidase, that is pro-PO, and production of reactive oxygen species. This happens in human beings, and this happens in insects. And in addition to that, in case of humans, what are the additional innate immunity parts or cells are natural killer cells, which are exclusively present in human beings, not in insects. Interferons, which interfere in the multiplication of the viruses, and the inflammatory response in mammals. That is where we will be getting so much allergic reactions like in inflammation. It may be throughout body temperature rise, or some development of boils, or some reddening of the skin. These are the immediate responses for innate immune system in human beings. And in case of innate immunity, as I already told, no immunological memory is present, whereas it is present in adapted immunity and found in nearly all life forms, whereas it is found only in vertebrates. And in case of adapted immunity, which we will be seeing what is the major thing in human beings, it is cell-mediated, particularly T cell and humoral components. Humoral components here, in case of human beings, it is antibodies to eliminate pathogen or detoxification. This is about first we will see what is innate immunity in insects. In brief, I explained. Now we will see what are the constants. I will go only pertinent vocabulary I will be using. I may not go in detail. Many of you will be knowing. Once I tell the heading, you may be you can reconnect it to what we studied in case of microbiology course or anywhere. In insects, exoskeleton made of chitin forms the first barrier we have seen. Another is lysozymes. It is an enzyme that breaks down bacterial cells, and hemocytes are phagocytosis. Till Dr. Hoffman gave the how innate system functions, in our olden textbooks, we might have seen only phagocytosis in insects. We don't know anything about that. But how this is happening, this innate immunity, they have explained. That is the reason they got that Nobel Prize. Then hemocytes also secrete antimicrobial peptides that disrupts the membrane of fungi or bacteria. Coming to the in insects, like human beings, in blood there are several types of blood cells. As of now, in insects, only 7 types of blood cells are identified. What I have mentioned here in the red color, red fonts is these are from the insect cricket. Granulocytes, plasmatocytes, prohemocytes, coagulocytes, inocytoids, spirolytes. These are present in crickets. And in addition to this, there is one more cell, that is adipohemocytes. These are the 7 types of blood cells present in insects. they will be having different roles, and unlike in human beings, they don't have contribute in the formation of B cells or T cells. That is the major difference, which you will be seeing in the coming slides. Here, if you see, this is the typical in Drosophila. These are the functions of hemocytes, crystal cells through melanization, plasmatocytes for phagocytosis or nodulation, for example, bacteria. Lamellocytes, this can happen in encapsulation when a little bit bigger parasites are encountered. They will go for lamellocytes. This is how. And as of now, all these blood cells are not present in all the insects. In some insects, only 3 types, some insects 4 types, in some insects 6 types. But it is not necessary that all the insects have this type. Mainly in Coleoptera and Lepidoptera, they say majority of these things are there. And this is the position in case of Drosophila. And this particular slide shows how this hours after infestation, what is the percentage survival after infection by these fungi or bacteria. This is how this mortality happens after the infestation of this either fungi or bacteria. This is how they could develop by studying through the mutants, defensine, what are the actual chemicals involved, drosomycin in different mutants. They have identified the exact chemicals which are responsible for formation of the antimicrobial peptides. Broadly, what is happening in insects, it can be divided into 4 pathways in this innate immunity system. First one is, take for example, if a parasite enters into an insect, for example in Drosophila, then what happens is lamellocytes differentiation happens and they form a capsule around the parasite, either it is a larva or eggs inside the body, so that they won't allow it to emerge or feed further. That is how this is one system. This is through melanization and through lamellocytes, a sort of blood cells. Another is in case of fungi and gram-positive bacteria, there are 2 paths. One is tall pathway. This is the pathway they have identified for which they got Nobel Prize. In this tall pathway, the diff and dorsal, they are responsible for the formation of ultimately the antimicrobial peptides. Tall, initially, though this tall gene was identified long back in 70s only, for which one more Nobel Prize has gone, that is for identifying tall gene as the required gene for the embryonic development of this fruit fly. And when they worked out, this particular tall gene is also required in terms of formation of antimicrobial peptides. And another pathway is particularly for gram-negative bacteria. It is the immunodeficiency pathway, or otherwise known as IMD pathway, where protease cascade is present. Through relish, it will be forming antimicrobial peptide. This is the third pathway. Another pathway is, apart from these pathways, for viruses there may be additional pathways like JAK-STAT pathway, and RNAi itself may be responsible for viral mRNA degradation. These are the broad pathways through which insects are getting protected themselves from the microbial attack. That is innate immunity. And now we will see what is happening in case of vertebrates. The immune system of mammals is best understood of vertebrates. Innate defense includes barrier defenses, phagocytosis, antimicrobial peptides. Additionally, as I told earlier, in case of human beings, natural killer cells, interferons, and inflammatory response. These are the additional things which are present, which are not present in case of insects. This is the inflammatory response. Mast cells are involved in this by releasing histamines. Activated macrophages and neutrophils releases cytokines, signaling molecules that enhance the immune response. This is how this inflammatory response helps in this immunity. Inflammation can be either local or systemic. Barrier defenses you can make out. Barrier defenses include skin and mucous membranes of respiratory, urinary, and reproductive tracts, and mucus traps, and allows for the removal of microbes. Many body fluids, including saliva, mucus, and tears, are hostile to many microbes. The low pH of the skin and digestive system prevents growth of many bacteria. In case of cellular innate defense, how it happens? It happens to the phagocytic cells recognizing the groups like toll-like receptors. In case of insect, it is a toll receptor. In case of human beings, it is similar to toll, and they gave the name of toll-like receptors. They are involved in the cellular innate defenses. Coming to the another major difference in nomenclature, and their functions are also different. In case of human beings, a white blood cell engulfs a microbe, then fuses with a lysosome to destroy the microbe. There are different types of phagocytic cells. What we learn is like neutrophils, macrophages, dendritic cells, eosinophils. When you go for the blood test, all these are accounted to see what is the level of white blood cells for diagnosis purpose. In addition, in case of human beings, natural killer cells, these circulate throughout the body and detect abnormal cells, and by releasing chemicals, they will cause this cell death, inhibiting the spread of virally infected or cancerous cells also. They are natural killer cells, or in short, they are also known as NK cells. Many cellular innate defenses involve the lymphatic system. Yeah, these are all the phagocytes and their relatives in case of human beings. Coming to the where, suppose in our body, if you see where this different immunity system or parts related to immunity are involved, if you see, starting from adenoids near nose, tonsils, lymphatic vessels, thymus, below our neck, chest region, then spleen, lymph nodes in the different body parts. These are all the different immune system promoting parts in our body. Coming to the like insects, we have seen there is a antimicrobial peptides and proteins. What happens in human beings, if you see, peptides and proteins function in innate defense by attacking pathogens or impeding their reproduction. Interferon proteins provide innate defense, interfering with viruses and helping activate macrophages. That's how they will help in reduction of pathogens. About 30 proteins make up the complement system, which causes lysis of invading cells and help trigger inflation. And what happens? One of the news always scatters us is what happens if evasion of innate immunity by pathogens happens. Why it happens? Some pathogens have a destruction of modifying their surfaces to prevent recognition, or by resisting breakdown following phagocytosis. Tuberculosis is one such disease and kills more than 1 million people a year. That is one example. Here, if you see what in the coming 4 slides, these are the pathways identified in both in insects as well as the mammalians. What I have already explained. In case of invertebrates, it is a toll pathway. In case of human beings, it is a toll-like receptor pathway, TLR, toll-like receptor pathway. And in case of Drosophila, immunity deficiency pathway is present. This is like a mammalian. This is in insects. This is the example of how blood clotting happens in case of insects as well as human beings. This in case of human beings, thrombin, prothrombin becoming thrombin, and thrombin will making fibrin into fibrinogen. That is the mechanism. And though it is similar in insects, it is in case of insects through the phenol oxidase activation. Coming to this invertebrate antimicrobial peptide response, if you see, that is mainly from the fat bodies. 90% of the fat bodies are responsible for that. Hemocytes contribute 10%. Whereas in case of the mammalian antimicrobial peptides response, immune cells or mucosal, etc. surfaces contribute 100%. These are the different antimicrobial activities in insects as well as human beings. If you compare whatever we listened till now, if you see, what is the similarity, what is the dissimilarity? If you see, all the pathways are in case of innate, their pathways are similar, but the responsible receptors may be different or slightly related to what is present in insects, which is conserved in the evaluation. Suppose in receptors, TAL is present. In addition, in insect, immunity deficiency pathway is there, whereas in case of mammals, this is TAL-like receptor is present. This is transcription factors are NFKB and IKB. This is both are same. Coagulants and immune cascades, it is mentioned here. Finally, if you see what are the antimicrobial peptides present in insects, example is defensins, secropins, moricins, gloverins, and atacins. Whereas in case of human beings, they are identified as defensins, which is common in both insects and human beings. Dermicidin, this is present in case of human beings. And coming to the proteins, antimicrobial proteins, if you see, lysozymes are present both in human beings and insects, which are required for the lysis of even bacteria. And sarcotoxin in case of insects, histatin, it is present in human beings. This is but broadly the pathways are similar to each other wherever they are present, with changes in the peptides or proteins or some receptors. Now we'll see a little bit on what is adaptive response. Adaptive response or adaptive immunity is happening through lymphocytes or white blood cells. When this is mainly through the T cells or B cells. Easily how you can remember is the cells, lymphocytes originating from the thymus region or can be called as T cells. The lymphocytes which are coming from the bone marrow, they are called as B cells. That for remembering purpose. This then what is their duty? From here starts the duty of identification of antigens, formation of the antibodies, and whatever we are discussing regarding so much thing about these vaccinations, everything will happen. That will cover in the discussion part. But slightly, I will go ahead with what are the different nomenclatures we will come across on daily basis when you are seeing this newspaper or some medical doctor reports are coming. What are the things we are seeing? We'll see the headings. One is the antigen receptors. They are from the both mature B cells as well as mature T cells. These B cells and T cells have receptor proteins that can bind to the foreign particle, whether it is virus, bacteria, or whatever it is. Each individual lymphocyte is specialized to recognize a specific type of molecule. And this B cell antigen receptor is a Y-shaped molecule with two identical heavy chains. This I will not go in detail. This is mainly antigen antibody formation and how this B cell do their duty. That part we'll see. This from these B cells gives rise to the cells that secrete a soluble form of protein called as antibody or immunoglobulin. Then 1 minute, please. B cell antigen receptors and antibodies, these are the things, this epitome which is present on this pathogen and which is linked with this B cell antigen receptors. Then from there antibodies comes. Then antigen receptor specificity, because it is required for each antigen, each type of antigen, there should be a different type of antibody. It may be A, B, C. This is how this antigen antibody formations happens. Then in the next step, antigen recognition by T cells, it happens. The tips of the chain form a variable region. The rest is a constant region. And here what we have to remember is T cell and B cell antigen receptors are functionally different. In infected cells, MHC molecules bind and transport antigen fragments to the cell surface, a process called as antigen presentation, which is very important in terms of identification of these foreign pathogens. Then another important thing in immunity system is B cell and T cell development. The adapted immunity system has 4 major characteristics. Otherwise, we'll have at one point of time in our life, we'll have some or the other problem. What is that? This B cell and C cell development should happen through diversity of lymphocytes and receptors. Unless these receptors are different for different types of microbes, that heterogeneity cannot be maintained, there will be a problem. Another is self-tolerance. Our immunity system should not attack our own cells. That is the important point. Another third point is B and T cells proliferate after activation. They should multiply in sufficient numbers. And most important thing in adapted immunity is immunological memory. Once some microbes have entered, through antigens, antibodies are developed. Next time, after few years, if some same pathogen comes with same antigens, our body should be perfectly identify and attack immediately without taking any lag time. That is the major difference in case of adapted immunity, what we'll be having. Then this is the example of a slide showing a generation of B and T cells. So origin of self-tolerance, proliferation of B cells and T cells. I won't take much time here. Once activated, a B or T cell undergoes multiple cell divisions. This proliferation of lymphocytes is called clonal selection. Two types of clones are produced. One is short-lived, known as effector cells, and that act immediately against the antigen, and long-lived one is known as the memory cell. This is what our antibodies for COVID, they will be doing their duty. When next time it comes, we need not to worry because it is already saved in the memory chip. That is what we are trying to have through vaccines now. Then immunological memory, if you take, initially antibody A will be there. After some time, its titer will be more. And suppose another antibody comes, its titer will be low. As the time progresses, this titer will be more for any antibodies. Here, adaptive immunity defense against infection by body fluids and body cells. That is the duty of this immunity. Acquired immunity has two branches. That is humoral immune response and cell-mediated response. In the humoral immune response, antibodies help to neutralize or eliminate toxins and pathogens in the blood and lymph. In the cell-mediated immune response, specialized T cells destroy affected host cells. Here, a type of T cell called a helper cell. This is very, very important. If this particular T cell is damaged, our immune system will be hampered. That is where some problems like deficiency of immunity happens, inviting so many diseases. This is the cytotoxic T cells are the effector cells in the cell-mediated immune response. This shows the activation of B cells. Different procedures in neutralization, how they will help in neutralization, opsonization, activation of complement system and pore formation. This is how this defense happens. This shows the different types of different forms of the immunoglobulins, IgD, MgA. Coming to in a short form, summary of the humoral and cell-mediated immune response system, if we have to remember. Both humoral means body fluids, and cell-mediated responses can include primary and secondary immune response. Memory cells enable the secondary responses. Here, this immunity can be broadly, it can be active or passive also. What is active immunity? Active immunity develops naturally when memory cells form a clones in response to an infection. It can also develop following the immunization or vaccination, COVID vaccination. This is how we can remember. Another is passive immunity. It provides immediate and short-term protection. It is conferred naturally when IgG crosses the placenta from mother to fetus, and when IgA passes from mother to infant in breast milk. It can be conferred artificially by injecting antibodies into a non-immune person. Here, if you see in the last 2 days, there is a attempt to give the two types of antibodies. They are calling it as hybrid antibodies for treating the COVID patients. This is also possible. They are trying. Then this is antibodies as a tool. This is innate adaptive. Just it is covered. Yeah. Here, whatever I told, I'll take 1 minute to sum up, because this particular slide gives whatever we discussed till now. It is giving like what are the different cells in the body which are helping in immunity. White blood cells, it can be neutrophils, basophils, they have different duties like engulfing and inflammation. Basophils helps in inflammation. Destroying of the homes is done by eosinophils. Monocytes do engulfing. Lymphocytes, specific immune responses will do. What are different lymphocytes? Lymphocytes are either B cells or T cells or helper T cells or NK cells. They are the natural killer cells, kill cells with the guidance from the antibodies. Here what happens sometimes when in this evolution, when our immunity is developing, these pathogens are trying to play tricks and make our immune system weak. That is here one best example is helper cells which are meant for our protection. They have been hijacked by these viruses, and the example HIV virus, when it takes this helper T cells, immunity comes down and making so many diseases will be susceptible to several diseases. This is the vaccination. I need not to tell a single word here. All of you are experts by now. Then what is the immune rejection? In case of human beings, that is possible. It is immune rejection is possible. That is why we have to be careful while giving the blood, because the same group of blood is to be given. Otherwise, it will be rejected. And it is to be kept in mind in while tissue and organ transplant, because in emergency cases in medical, they will be giving suppressants, immune suppressants. Then they will be doing these surgeries. Then allergies, sometimes extreme allergies are there. Sometimes they can be fatal also. While taking these injections, they are asking, do you have any allergens? Normal things may not problem. If somebody has never taken injection, if a slight thing also gives an allergen, that is to be informed before taking vaccination of this. An acute allergic response can lead to the anaphylactic shock, a life-threatening reaction within seconds of allergen exposure. This is a rare case. Then there is an example of autoimmune diseases. Autoimmune diseases is individual with this type of disease, immune system loses tolerance for self and turns against certain molecules of our own body. That is the problem. This is what I want to share regarding this thing. Coming to the how to make our immune system more, how to be more immune. If you see, the study shows exertion, stress, and the immune system. Exertion and stress, they will diminish the immune system. There are sufficient studies over that. Dr. Mandakranta can explain regarding that. And immunodeficiency diseases. Example is that helper T cells I told. If that comes, acquired immunodeficiency syndrome happens. That is how what happens here. In case this load of HIV titer will increase gradually over the years, and whereas the helper T cell concentration comes down. That means our immune system is being completely hijacked by that virus. That is where we have to keep it down and increase our helper T cells. And same is the case in case of cancer also. Many of the cancers, the frequency of certain cancers increases when adaptive immunity is impaired. 20% of all human cancers involve viruses. That is where we have to be careful. This is the antigen variation. What are the common things we have to keep for building our immunity? If you see, it starts with our hand washes. Our sleep cycle should be very good. Minimum 7 hours of sleep, and nutrients rich in food. In horticulture, we have all total this fruits and vegetables starting from A, B, C, D. Of course, you will get freely from sun, and minerals like iron and zinc. Cortisol levels, which is a stress hormone, we have to keep it down. Then supplemental intakes, exercise, and happiness. These are the things which will boost the immunity. There may be still studies are required. All these things definitely they will build the immunity that will. And for happiness chemicals and how to hack them, it is mentioned here. Happiness chemicals like dopamine, oxytocin, serotonin, endorphin. They are by good activity, positive thinking. These are all can be we can have for good immunity. This in recent studies they have shown from ISA Foundation, they have shown that immunity through inner engineering and meditation and yoga. They have seen, and that is in publication stage, this has increased the immunity level in the human body system. Many things are still to be studied regarding understanding and increasing immunity. Immunity protects us, we need to boost it. That's all, that is the requirement from our side. Be immune and be safe. Thank you. Thank you. With this presentation is completed, and we will go with the discussion. Because it is two systems, I was little fast, and I have given a glimpse of the idea. What is present in case of insects, what is there in human beings. And now I request all the audience to participate and share your experiences regarding either in entomology or if it is related to human being, this immunity. Dr. Mandakranta also will join for answering. Thank you. I invite anybody can start this one.
Dr. Sridhar, to set the discussion going,
Yeah, please.
I'll ask you a little funda. Are there any natural diseases of cockroaches? And how is it that cockroach evolutionarily is so effective in warding off any kinds of pathogens or anything? That has anybody gone into the deeper molecular mechanisms in cockroaches and their ability to survive all kinds of pathogens?
Okay, so cockroach I didn't come across. If it is there, I will refer and I'll let you know by evening.
Because the
majority
reason that I'm
studies,
asking also, is in order to understand, we might have to understand how the tough one is managing everything.
Right.
Because it literally lives in the gutter, literally eats everything, and in spite of it, proliferates like anything. Like
that is true, sir. Cockroach, except light, it fears. It won't have fear for anything. Only light, only it doesn't like, I think.
Because in the case, if you draw parallels in between a mice and a human being, if we can make 10 to the power of 9 different types of antibodies for 10 to the power of 9 proteins, it is said that mice can make up to 10 to the power of 13 or 14 different types of proteins. So that explains how they are able to survive and ward off all. It will be very nice to see some of the things, because it's over 400 million years of evolution. They have both acquired as well as the tremendous ability to survive, and they probably will see. Many times we are talking, again, as I said in the previous, in general pattern of immunity or resistance, whether it is, but the famous geneticist said that I don't believe in God, but if God were to exist, he certainly had a passion for beetles, which form the majority of insects. So he's from Bhubaneswar, so you should be knowing much better. So having said that, I am only asking that 75% of the living things are insects.
Insects.
And each family and each order has resistance or development of resistance to a particular pathogen does not follow. It is multi-directional and multi-pathways. It's not always uniform or across. So in many times, it will be nice to select certain groups of insects. For example, cockroaches is one classical case. Maybe the same thing that applies to bed bugs, which are emerging as major problems. Houseflies, which literally live in filth and don't seem to get infected, so those things will add a lot of. The advantage of it is the insect physiology and human physiology are very much alike, with a few exceptions. So that way, there are a lot many things to be understood. If you can learn from heterorhabditis and photorhabditis, then we have a lot more to learn also from insects. And I think we have not understood the immunity response in different groups of insects. The response of a parasitic wasp is different. For example, I am fascinated by the gall midges, which with a micro dose of a chemical can induce such big galls and plants.
Yeah.
So there are many things. I was only thinking that whether any deeper studies, I'm sure in medical field, certain studies could have been done. And even within the cockroaches, there is Periplanata americana. In fact, I don't know whether many of you know, this Blatella germanica, the name Germanica was given by Linnaeus because he was a Swedish taxonomist who hated Germans. So he called it as Blatella germanica. So the thing that I would like to say is, if there is one thing that the humans hate, it is cockroaches, for reasons whatever it is. I think there is a lot more thing to understand from flies, cockroaches, and for example, Culex. It breeds absolutely on filth, but nothing seems to be really, except some predatory fishes and other things. I don't think there are. Maybe there are certain things that are involved with it. Any lights on specificity associated with insects and how they are able to overcome?
Specificity is, suppose, take for example, Bacillus thuringiensis. Is good for mosquitoes, Bacillus thuringiensis, other species for lepidopterans. That way, different strains with different insects, that is there, sir. That is there. And one reference I have seen that, suppose in case of Beauveria bassiana, we have been thinking that there is no adapted immunity. But whether is there any possibility of development of resistance to antifungal? Because luckily, gut pH is deciding the efficacy of this BT formulations. So that is the reason there is a specificity. With the over the time, it has to be studied. But a lot, as per the reviews also, what they say, more than what is studied or observed, lot more to be observed in different insects because they have been conserved, they have been surviving. What is happening to be studied.
Dr. Mandakranta would like to add on cockroaches and houseflies.
Sir, actually, I found it very fascinating, this discussion, because we don't realize how robust an immunity God or whatever life has given to these smaller forms as compared to them. In fact, we are a very fragile organism. That is, and I really appreciate the way Dr. Sridhar has exhaustively covered immunity in such a way it was better than a microbiologist as far as I can say. And he gave some very pertinent points, especially on some important things. Like, we never take this in our clinical practice. Actually, till COVID came, I don't think any general practitioner actually thinks about the B cells, T cells, or the lymphocytes. But finally, it has come down to that. And if you were to compare with the innate immunity, which any organism, any of these, like you said, cockroach, for example, it is wonderful the kind of inbuilt immunity they are having. We rely more on our adaptive immunity. On top of that, as you can see, because of our sterile environment, because of the way we have evolved, human beings, we are more, in fact, we rely more on artificially induced adaptive immunity in the form of either in the form of medication or in the form of vaccination. And I think over the times, in the millions of years where we have been evolving, it is definitely these lower organisms are a notch above human beings. The reason why we have survived, I would say, is because of our cerebral capacity and not because of our innate immunity. I didn't get the actual term you were asking, sir, regarding the organism and humans. I mean, I don't know. If you're comparing the immunity, definitely we are way behind. We have to develop some kind of, that is why how we are surviving any disease is basically manufacturing a vaccine. Our own innate immunity has been declining because of our lifestyle changes. Like you already know, diabetes and cardiovascular diseases make us more prone towards a reduced immunity. Various factors are involved. But basically, he has covered so well about the cellular level and the humoral level. Humoral level meaning all these antibodies which we develop. All these are suppressed because of the lifestyle we lead. Now, this is so it is at such a level that we cannot define it. It is more at a molecular level, and I would be definitely not an expert on that kind of a thing. But yes, finally it comes down to we rely more on artificial adaptive immunity rather than our natural innate immunity. It exists. Of course, we have been made into a wonderful organism. Otherwise, all of us would be dying from every single bacteria or virus. But why we don't is we know that it exists. But really, COVID has changed equations to such an extent we cannot explain. We can't explain why our innate immunity is not sufficient, why our even our adaptive immunity is it falls short for a small thing like a virus particle. We are not able to prevent its replication within the body.
Good.
Actually, Dr. Sridhar had just asked me to interact, so I have not prepared anything regarding it.
The idea
This is what I was
is
expecting.
to make more persons participate and give their differential. Sridhar, the specific question I asked with regard to cockroaches and houseflies was, in the overall scheme of immunity, the role of symbionts associated with each of these in the plays a tremendous role. I think the key that the real key with regard to the survival of whether it is termites or whether it is cockroaches or houseflies lies with how well we understand the endosymbionts that are associated, not necessarily in the gut microflora, but even beyond that. So I was only thinking of looking into a more deeper understanding of the contributions of the symbionts in conferring, because I don't think an individual entity by itself is able to come out, but their gut microflora with which they are associated and also in the other regions, and what is their role in developing that kind of a thing, either through biochemical pathways or through prevention. I think it's something that needs to be probed much deeper. Otherwise, the evolutionary closeness with which a symbiont is associated with the microflora of a particular organ or organelle cannot be that easily understood, because without those microflora, they are or the symbionts, they just seem to die just like any other organisms.
Right, sir. This is an internal biodiversity then in our terms.
Internal diversity and ecological function. Chandish, you cannot afford to remain silent.
Yes, sir.
Hello, sir. Hello, Sridhar. Hello, Manda.
Hello.
Very interesting talk, Sridhar, especially trying to draw the parallels between insects and human beings, specifically on immunity, where you don't want the insect to be immune, and we are trying hard. We really don't want insects to be immune to fungus or virus, or because that's how we are trying to manage. It's not a question. One thought which came to my mind, one very interesting study which I had come across on Oryctus rhinoceros. A virus, Oryctus rhinoceros virus, was used to combat Oryctus, right?
Yeah.
So one population in Guam was totally resistant or maybe immune to the virus. Yeah. Now, why we do not have a population which is immune? Why that, or haven't we looked at it that way? That's one thought which was coming to my mind, because it was 99% successful, the Oryctus virus, except one population. So it meant that strain of Oryctus was tolerant or immune to Oryctus virus.
Specific receptor may be absent, and for the virus, whatever is there.
It's the same species. See, Dr. Krishna Kumar was mentioning about different species of insects being more susceptible to a particular fungus, or but this is the same species, a different strain, probably. So this thought came to my mind. That's very nice, Sridhar. Excellent.
Okay, thank you. Thanks.
Last year or before last year, I was in Samoa for International Coconut Conference, and this particular aspect came into the aspect discussion of the varieties from Guam Islands. In fact, it has become a serious problem, and they are not having this thing. What do you understand by, of course, it's this phytoplasmas and relationship with the vectors? Do you think they are pathogens? They are pathogens of or they are just, what is your opinion with regard to their association? And why is it that the plants are not able to develop any immune response to that?
It is maybe parallel to these viruses. Whatever problems we have with virus, they what we are telling, like
they are more similar to bacteria than viruses.
Right. Okay.
I thought, because these are all emerging problems, but
right.
Does the insect, when it is transmitting a virus, does it fight the virus?
Fighting virus when it is multiplying inside the body, because it is persistent only, it is possible in that.
No, there are many questions. There are no easy answers.
Yeah.
There appears to be, but there is no 100% proof with you cannot hammer it and say that yes, Anopheles, something replicates in Anopheles, but the mosquitoes efficiency doesn't seem to come down. The other aspects which I would really look into based on some of my own studies is, we study a particular individuals or 10 or 15 individuals and come to a very broad consensus. But that does not happen in nature, because we are not looking the population through population genetics. The individuals within a population are grossly varied. So just because something is banned, whether it is resistance, susceptibility, transmission, or whatever it is, it has to be viewed from a function of not just selected samples, but how the population at large responds to the pathogenesis, the resistance, and how it carries forward. And so far, we are looking everything from our perspective, but we are not looking from the pathogen perspective. And the classical case is the influenza virus. See how they are able to come out with a very beautifully changing their coat proteins and their other things in order to adjust themselves to better infection or better survival. So some of these things need to be looked from not just the biochemical mechanisms by with the, like he said, Dr. Sriram, about the coevolution. But beyond coevolution, there is also reciprocal coevolution. The reciprocal coevolution is one which overcomes a particular resistance and has a selective advantage, like a Parthenium beetle. So those kinds of reciprocal coevolutions also operate in nature, and those that are successful in reciprocal coevolution are some of the most successful species. So my only purpose in drawing all this is, while we are going too much into molecular and biochemical and other aspects, which is good, we are missing on the very important ecological bus.
Yes, right.
This is what I thought I'd share. Sriram can add if he wants to. He has all rights to differ with me.
I agree, sir. Actually, biodiversity, in many papers, finally they say biodiversity is the key to have the different, to bring the resistance sources.
See, biodiversity, what it does is not resistance or susceptibility. The very function of biodiversity is to keep the system under equilibrium. When the system is under equilibrium, whenever there are small hiccups, then what happens is the biodiversity is robust enough to quench those kinds of upheavals and again stabilize into that kind of a level. That's what happens whether it is with our the symbionts that are associated with inside stomach, or for that matter the biome associated with the roots. Any small hiccups that happens, these associated organisms, they nullify the indirect effects or the negative effects and again set the equilibrium on site. The second one is biodiversity doesn't allow a particular species to dominate over other, thereby again maintains an equilibrium. And that has to be understood both at the micro and at the macro level. So I think a lot more investments has to go in ecological biodiversity at the micro level than just probably planting few trees and telling that we have contributed to biodiversity.
Right. Okay. Okay.
Anyhow, I think
Thank you, sir,
I
for
want
your
to talk anything I would. Kamala Jayanti garu unnara?
Sir, good afternoon, sir.
So is it how strange it is we have to probe everybody to speak?
No, we love to hear you, sir. After a long time, we are hearing you. Thank you for such a wonderful interaction.
If you want to spoil somebody, you praise him.
No, sir.
Huh? Veena? CTCRI? This problem with virtual is they put their, presence and then they can disappear. That's okay.
All others are requested to share.
Whoever wants to add a note. Sai Prasad is who? Sai Prasad?
Who is in ITC presently, sir? Who was in?
Yeah, that's what I thought. Sai Prasad garu unnara?
Yeah, sir. All well.
How come Sai Prasad is silent? Because Sai Prasad.
I am just hearing. I am away from your pathology and entomology, so I am just trying to how to correlate to. I could only give him certain analogy on how human immune system works to Sridhar a little bit when he has called me on this, how he can compare with that.
Yeah.
True.
Otherwise, all well, sir. How are you?
I am good, but what does the ITCs throw into this whole thing in understanding and making it with regard to this? You make shampoos, but also you should come out with something on immunity.
Yeah, we are coming now. We have a lot of our products which are coming up in health and wellness area, sir. We are coming into the foods where you have a lot of products which we are trying to make in the coming days. Of course, we already have few of them, sir. Like you have immuno jellies for kids, where we have some vitamins and like vitamin C and zinc into those products. And also we are also coming up with some of the food products where you have, of course, it's all in the digestive range in the biscuits and all those, sir. But in the coming days, you might see much more in this area.
Sai Prasad, thoda ITC te hatke boliye. Are we overplaying antioxidants a little too much? You can speak away from ITC for a moment.
No, nothing like that. I think they are really beneficial, sir.
So they are beneficial, but it has taken such a commercial angle where benefit have gone down the rock bottom and only commerce has surfaced.
I don't think so. Actually, people are making using it for the commercial scale and getting gains by putting them into products and claiming that way. I don't think it is the right way to do it. But if you have a proper claims and then there is a real scientific data behind what you generate in the products and then the level which they needs to be in the product so that it will give you that appropriate benefit, I don't think people can make. I think nowadays, including consumers and everybody regulators are up to it, sir. You cannot overclaim certain things where you don't see any benefit in that, and then you might put in your anxiety and then put in your product and claim too much on it. If you are not able to withstand to it, I think you'll put reputation on your company or product, and I think everything will go down. And that is where you can believe in ITC side. It will be very strong in when we put certain claim, where we are doubly sure that what claim we are making and what is the benefit we are talking about when we are putting in a product and claiming that, sir. I think we stand to that, I believe.
1:1 మాట్లాడతానండి.
I don't know, 1:1, sir, but I brought that up.
I talk to you, not on this, on something else.
Sure, sir.
Thank you, sir. Thank you, Sai Prasad. See, the purpose of probing this is, somebody like Kamala Jayanti, Veena, you at least should add up to the, science of the discussion. That's the purpose with which I am probing. Subharan?
Yes, sir. I have a query, sir. It's slightly medical and entomology oriented, and it has been going on in my mind for a long time. With the black fungus coming up, amphotericin B is the drug in line. Now, there is something scary about amphotericin B, though it's going to do good, because in a journal of Eye published by Royal Ophthalmic Society, there was a paper long back in 2018 where they have told that the use of metarhizium anisophilae, it's a freak case, not continuous, did cause ocular keratosis. And in one of their studies, when they had done that, they said that it is susceptible to some of the natamycin or something, but it is highly resistant to amphetamine.
Subhaharan, you're online.
Okay, there is a call drop or something.
Yeah, there is connectivity problem. Once he
Hello.
comes.
Yeah,
Hello.
I'm sorry, I
please
lost
continue.
contact. Yeah.
Please continue.
There was a report from a medical journal, from Royal Ophthalmic Society, where they have noted a free case of ocular keratosis caused by metarism anisopyle. And the line of drug that was used was they had used natamycin or something, but they have also screened various other drugs and told amphotericin B is the most resistant stuff. I mean, the fungus is most resistant to amphotericin B. Now, this amphotericin B is the first line of defense as far as the black fungus is concerned. Now, in a situation like this, now if there was an exposure and that the metarism was building up on an ocular keratosis, what would be our say on that? It was just a thought running in my mind, which I thought I could ask maybe Sriram or any medical fraternity or anybody.
Dr. Mandakranta, anything?
He was referring to amphotericin B being resistant to treatment of keratitis, which was due to a fungus. Was
yes,
this specifically
No. I'm sorry. My query is not clearly understood. The issue of metarism anisopyle, which is used as a myco-insecticide to control insects, did cause ocular keratosis in few of 3 cases in many countries. And when they had tried to do the corneal scraping and find out what the fungus was, it was established that it is metarism anisopyle. Then the line of therapy that they had used was natamycin or something. But during the process of evaluating various drugs, they found that this particular fungus, that is metarism anisopyle, was highly resistant to amphotericin B. Now, with this mycorrhizasis on rise and amphotericin B being used as a first line of treatment, if accidental exposure of this metarism anisopyle happens, and if it was to rise, it is just a hypothetical question. It's not that.
Okay. Anyhow, as of now, metarism is not a problem for us. By chance, if it happens,
if it happens, because it has been reported to cause ocular keratosis. How prepared are we? It's a hypothetical question, and the mode of preparation where we have to maybe initiate some discussion and also keep the medical fraternity well informed about things. It was just a hypothetical question. It's not that it has occurred, but in case.
Generally, any antibiotic, they are very site specific in action. So whenever we develop any fungicide for any either medical purpose or in the agriculture purpose, whenever we are developing single site mode of action for molecules, we have to expect the resistance to that chemical. Fungicide resistance will definitely happen. So the alternative chemicals should be in pipeline from the industry with a different mode of action.
Different mode of action.
I totally agree with you.
I agree that now at present we don't have. The alternative chemical has to come from the industry. Antibiotics production, it should be there. Alternative antibiotics should be available for this fungus.
No, Dr. Sriram, my question was not on the alternatives being ready. My question was that given the situation in the table, now the medical fraternity should be well informed that because this fungus did cause in a freak ocular keratosis, now this is a thing which you should look for.
Same way, there are many incidents of infection by biocontrol agents, opportunistic or immunocompromised people. Opportunistic pathogens in reports are there, but they are very, very few in number. And in many cases, it is the immunocompromised person, they succumb more, as now the steroid is playing. So we have to be very careful.
Yes, Dr. Sriram, I said, no, it's not a regular occurrence. It's a freak occurrence which has been noted and which that's what I was coming to say. Not that it's a regular occurrence. It was just a freak occurrence, but we have to be prepared for it.
Thank you. Noted that one. Thank you.
This is Sridevi.
Yeah, that's right, Kumar.
This is Sairam Kumar. This is Sairam Kumar, Professor Entomology from Bapatla and alumnus of IRI. So I just would like to add a point here to Subharan's viewpoint. In a single case study, how can I don't know about the authenticity of this publication, but he himself has been telling that it is a single freak incident wherein metarhizium has been reported to be highly resistant to amphotericin. There are 24 alternative drugs recently have been found against black fungus. Osaconazole is one of them. I don't know how this study has been taken place, but anyway, it is a freak incident, he said. Okay. My point here to the topic presenter here, that's Sridhar. I've already given my question in chat box. Please go through that. And then there are several incidents. I absolutely agree with Dr. Krishna Kumar, sir, who has given a real insight into the insect immunology or whatever. So there are so many incidents, so many historical literature. If you go through in silkworm and bees, honeybees, there are been several efforts have been made now to grow resistant or tolerant clones or breeds against several bacteria or viruses, and then we are producing several of the beneficial insects for the welfare of the humankind. So we have to really look into the insect immunology. I really back if Dr. Subramaniam is in the group. He used to teach us very good in insect biochemistry and used to teach us the insect immunology also to some extent while we were in IRI. So this is what I just like to add some points here.
Okay.
So I have been trying some studies on immunoproteins in some of my works here. So slowly we have to try upon lot of antimicrobial peptides are there. Lot of things have to be still done, and we are still the nascent stage, I think.
That is true.
A lot of work to be done.
Okay. Thank you. Thank you.
Sir, this is Sridevi. Excellent talk, sir, by Sriram sir, Sridhar Garu. Thank you for that. And then also thought-provoking discussion by Krishna Kumar sir, Chandish madam, Mandakranta madam. I enjoyed the talks today. Thank you. I just want to thank Atme and the speakers and discussion.
Okay,
thank you.
Dr. Meera Pandey is there? Dr. Meera Pandey? She must have left because 60% of the mushrooms are only for the medical field.
And she has left.
She has probably left. I am asking a question which is simply out of the box.
Okay.
How should our immunity response be with putative engineered microbes?
Yeah.
In future, you are going to get a lot of microbes which are going to be engineered.
Engineered, yes.
And those are very cleverly engineered to overcome all kinds of resistance, as it has been speculated now. Let us not come into any kind of a conclusion.
No, somebody has to become a reverse engineer then.
No, what I only meant is, as Sriram said, bioinformatics is one such tool which is going to be extremely useful in artificial intelligence, as well as in understanding the psyche, as well as coming out with a kind of repertoire with regard to the kind of mechanisms that need to be undertaken by simulated programs that can operate. I feel that this is one area which is going to become more and more applicable in the days to come, considering we are living in a world which is not everything is black and white. There are so many gray areas that are going to operate, and that also calls for preparedness as well as drug discovery, and also the only thing that the whole thing has exposed is our absolute lack of understanding of the factors contributing to the epidemiology. You can have a lot with regard to many of these things, but you should also know with regard to the pathogenesis, the epidemiology, the weak links with which you can attack a pathogen. For example, Staphylococcus is not a very ordinary kind of a bacteria, but Dr. Mandakrantam would support me that the kind of resistance that it has developed to n number, probably you will be running out of any kind of antibiotics before it totally comes out. And it is even speculated that in the days to come, with our ways of living, of hygiene, and more, if a Staphylococcus infection occurs, there may be no chemical or medicine that's available across the shelf to fight. So these are, and such organisms are very easily can be manipulated to introduce into hospitals and such other system, and you can set up a devastating cascade of problems with which you will have no answers. So my
bioterrorism, it comes to.
Please go ahead, madam.
Sir, I was just saying it comes down to bioterrorism, because that is one of the theories which is now floating around regarding even this present pandemic.
Yeah.
So, like what you say, in fact, is such a scary situation. If there is so much of research going on as far as replicating a virus by tweaking its genes or by tweaking the RNA, you are going to get again pandemic after pandemic coming if there is no control over it. Yeah. It is a very black area, in fact, of research.
One is tweaking the genetics. Another one is very, very stringently selecting a resistant population that can overcome all kinds of antibiotics. And so nobody will accuse you of genetically engineering and releasing it to something. But somebody can, so, I mean, these are all things I don't know how prudent it is to say. But at the same time, my purpose in making this statement was a lot more investment has to be done in the case of the bioinformatics. Ultimately, it is the tertiary structure and the coupling mechanisms which really determine one is able to establish within a cell or not able to establish within a cell. So the tertiary folding holds a key to a number of things, yes and no. And that can be very beautifully addressed in the today's bioinformatics. So I was only, from a theory to practice to where exactly we should be moving, whether it is a plant or animal, or with regard to some of the things that we really have to look is, in many of these things, we have not looked into the intermediary of animals, zoonotics, which are really going to play a very big role, because it is no more a backyard poultry, or it is no more a cow or a pig which is there somewhere roaming around. It is now everything is an organized kind of a packaging, like sardines, everything is in a very structured way. It only takes a bubble to burst before the, like H1N1, it is going to spread so rapidly that before your immunity, you think about what is happening, it would have taken a toll. So the addressing these kinds of things requires a number of other things with which we may have to address this. And one such is also the addressing the aspect of immunity. So the purpose of it in telling is, again, stress more on ecology, stress more on epidemiology, stress more of anticipated research. And those kinds of things will contribute probably with regard to an understanding of many of these things.
Right, sir. Thank you, sir. Anybody else is there to speak on this aspect? Dr. Patil is there. Dr. Patil, sir. Dr. Varghese.
Manmohan is there.
Dr. Manmohan. He joined, sir. Afterwards, I didn't see. Dr. Manmohan.
Okay, I think if it is Kamala Jayanti garu emana cheptara?
Everything discussed, sir. Everything discussed.
Any feedback from the fruit fly syndrome?
Yeah. No, sir, nothing.
Okay.
Just like that. Thank you, Sridhar, for a good discussion.
Okay. If and last final call, if somebody would like to express regarding anything regarding immunity, you can do. Otherwise, I profusely thank for this.
One more small question, Sridhar, to Dr. Mandakranta.
Okay.
Repeatedly sanitizing our hands, are we making it more and more, allowing the pathogens to become resistant?
Sir, there is a terminology which we use in a very flippant way. It is called the Pani Puri syndrome in India. In fact, when the first wave hit us and we had a very, very mild scenario in India, one of the factors we said is we are protected by our innate immunity, which we have evolved because of poor sanitation in our country. In fact, I remember there were some people who said, yes, too much of sanitizing may in fact make us more prone for any kind of a viral pathogenesis. Clinically, it is actually theoretically sanitizing is important. If you want to avoid entry of any kind of bacteria or virus, assuming your immunity is not sufficient to overcome the disease. But again, what you say, yes, over sanitizing ourselves is not good because that little bit of pathogenesis, the exposure to pathogens is actually what also builds up our immunity. The more your body has some kind of an exposure, the more your normal barrier is exposed to a virus or bacteria, the more chance you have of fighting it actually. So, which is why when first the pandemic started, everyone said it's going to be like a flu. It is going to be like flu. Some people will get severe, some people will just have a mild illness. But when the second wave hit us, we are not sure where the immunity went.
Just imagine for a second, if a pathogen can overcome the sanitizer, what will be our present status? This is what I was driving home the point. Sridhar, sorry for phone lo unnar kada.
Please tell me, sir.
No, you can go ahead with the.
Okay. With this, I think everybody is through in terms of their contribution. I profusely thank all the participants, particularly Dr. Krishna Kumar, Dr. Varghese, Dr. Patil, and Dr. Chandis Madam, that's Sai Prasad, Dr. Manmohan, and all other my colleagues from different institutes also. Each one of you, I thank you so much for attending this monthly talk. And now next month we'll be meeting with a new talk on a new topic. Till that time, I say goodbye to everybody. Thank you, everybody.
Thank you, sir. Thank you all
for joining and clarifying the medical doubts.
And give a big hand to
Sriram for initiating betting and immunity.
Thank you, Sridhar, for a good talk and a good discussion. Thank you.
Okay. Thank you so much, sir. Thank you.
Thank you, sir.